This video is going to provide an overview of the process of DNA replication. So remember, DNA is the genetic material. So all cells and all living things, which are made up of cells, must have DNA. So it's really important that they have a quick and efficient and accurate way of copying their DNA so that cells that are being made will have a copy and they can function as they should. So what we're going to do is take this rather complex and abstract process and kind of break it down bit by bit. So with this video, we're going to focus on just big picture, what's going on. In the next video, we'll kind of zoom in a little bit more, get a little bit more detail, and then we'll zoom in even farther and get all the things, okay? So what we start off with for DNA replication is a parent DNA molecule. That's the DNA that is already in the cell that needs to be replicated so that newly made cells can get a copy. So this parent DNA molecule is double-stranded, as you can see in this picture. And remember, the two strands of nucleotides are anti-parallel. So the top one goes 5' to 3'. The bottom strand is going 3' to 5', so they're just opposite. And then remember, the two strands are also complementary. So even though I'm not showing you the base sequence for each strand, you would know that they are complementary. So if there's adenine on the top strand, we'd have thymine on the bottom strand, so on and so forth. Okay, so we've got our two strands of nucleotides that are making up our starting parent molecule of DNA. So it turns out that in the middle of these two strands, holding them together, okay, are going to be hydrogen bonds. If you go back and look at the picture from the quick refresher of DNA video, and I showed you the base pairing, if you look between the two bases that are paired together, you'll see that what's holding those two bases together are hydrogen bonds. And remember, hydrogen bonds are the weakest type of bond. It doesn't take a whole lot to break those bonds. So during DNA replication, the hydrogen bonds that are connecting these two strands together are going to be broken, okay? And that's going to allow the two strands to separate, okay? So notice we still have our 5' to 3' strand. We still have the 3' to 5' strand as well. They're just no longer hydrogen bonded together. And of course, the bases would still be there as well. So by separating these two strands, we're able to use each strand as a template. So this is going to be very similar to what we did in the last video for the apply your understanding or apply what you know. And so if we have our 5' to 3' strand in orange that we started with, the cell can create an anti-parallel and complementary strand. So that's what I'm showing you in blue, the new strand that will be made. Likewise, that original 3' to 5' parent strand, the one here in orange that we started off with, it's going to be used to create an anti-parallel and complementary strand of nucleotides, okay? So at the end of the day, or I should say at the end of the process, we are going to have two DNA molecules. They're going to be identical in directionality and sequence, but notice that one daughter DNA molecule has one of the original strands of nucleotides and a newly made strand of nucleotides. And that's going to be the case for both of them, okay? So we call these two DNA molecules that we now have daughter molecules. So let's look at another animated visual of this. So in this case, I added in the base sequences. Okay, so notice our parent DNA molecule has two strands. The top one goes 5' to 3' with this sequence. Then the second strand of nucleotides is anti-parallel. It runs in the opposite direction. And if you look, base pairing is happening. So the two strands are also complementary, all right? Always got to repeat that to yourself. The two strands are anti-parallel and complementary. So remember, what I'm not showing you here is right here where my mouse is in the middle, there are hydrogen bonds that are allowing these bases to pair together, okay? So we have weak hydrogen bonds here that are joining these two strands together. So at the beginning of DNA replication, those hydrogen bonds are going to be broken, allowing the two strands to separate. So now we can use each of these parent strands as a template. What that means is we're going to use it as a guide so that the cell can build the anti-parallel and complementary strands for each of these, okay? So there's going to be a lot of clicking, so bear with me. So we're going to start with this top strand. So notice the top strand in my example runs 5' to 3'. So the new strand that the cell is going to build must be anti-parallel. So it's going to be 3'. And notice it needs to be complementary. So because there's an adenine on the top strand, the newly made strand needs to start with a thymine, right? Because they're base pairing. So then if we have guanine as our second nucleotide on this parent strand of nucleotides, what should we have next? Cytosine on our newly created strand. So with this blue one, y'all, the cell is building this, okay, using this top parent strand of nucleotides as a guide. So I'm going to go and just keep clicking through this, but notice that the two strands are always going to be complementary. So because our third nucleotide here is a cytosine, on our newly made strand, it's going to be guanine. Okay, so then we're going to have adenine, adenine, thymine. I'm just going to keep clicking now. So the cell is going to build this new strand one nucleotide at a time until it is finished. And notice now we have a complete DNA molecule. The two strands are anti-parallel, check. The two strands are complementary, check. All good. Now the same thing will be happening down below. I'm not going to do 100 clicks, but now in this case, because we started with the 3' to 5' strand, the cell is going to be building the 5 to 3. Okay, but notice it's still complementary as well. So if you compare these two daughter DNA molecules, notice they're identical, right? The top strand runs 5 to 3. Notice the sequence of that strand is A, G, C, right? So on and so forth. Same thing for the bottom strand. They are two identical copies of DNA, and we refer to them as daughter molecules. All right? So that's going to be the end of this just overview video. In the next video, we're going to kind of zoom in on the details, introduce some more vocabulary. So recommend you get some caffeine or whatever helps you focus, and buckle up. We're about to get into the details. Let me know if y'all have any questions.